Hello everyone, welcome to my YouTube channel, and today I will start off with a case before I'll present our lesson for our Human Anatomy and Physiology course. Let me start with a certain case all about a kid named David. David was 7 years old when the first symptoms appeared. His appetite at meals increased, and he always seemed to be hungry, and despite eating more, however, he was losing weight. And when he started asking for water instead of soft drinks, David's mother became concerned, and he wet the bed 3 times in a row. She knows something was wrong. The doctor listened to David's symptoms and ordered tests to determine the glucose concentrations of David's blood and urine. The tests confirmed the diagnosis. David has diabetes mellitus. In David's case, the disease was due to lack of insulin, a hormone produced by your pancreas. David was placed on an insulin injections, a treatment that he would continue for the rest of his life. And 100 years ago, David was would have died not long after the onset of symptoms. The field of our lesson today is concerning with a field of endocrinology, and you have an idea what is it going to be like. The study of hormones was then in its infancy, and most hormones had not yet been discovered. There was no treatment for diabetes and no birth control pill for contraception. Babies from inadequate secretion of thyroid hormone did not grow or develop normally. Today, all of these has changed. We have limited and identified a long and growing list of hormones, and these endocrine diseases that once killed or maimed can now be controlled by your synthetic hormones and sophisticated medical procedures. Although physicians don't hesitate the use of these targets, we are still learning exactly how hormones act on their target cells. And this chapter will introduce you to a new lesson, and this is all about the introduction to your endocrine system. You will learn more about the individual hormones, and as you encounter them in your study of the different or various systems in the human body. Let me start off with this quote. The separation of the endocrine system into isolated subsystems must be recognized as an artificial one, convenient from a pedagogical point of view, but accurately reflecting the interrelated nature of these systems. This was said by Howard Rasmussen in William's textbook of endocrinology in 1974. We have to, make this study more of an artificial one, okay, and it reflects the entirety of the system. If they try to look at the separation of this system. So before we move ahead with our lesson, let me introduce your learning outcomes about hormones, the classification of hormones, and many more. So we will have to explain 4 criteria that make chemical signal a hormone, explain what the cellular mechanism of action of a hormone is, what are the 3 chemical classes of hormones, and give an example of each, compare endocrine cells, synthesis, storage, and release of peptide and steroid hormones, compare the location of hormone receptors and the cellular mechanisms of action of peptide and steroid hormones, what are the 3 groups of amine hormones, and describe the role of the nervous system in the endocrine reflexes. Now, comparing the structure and the function of your anterior and posterior pituitaries, what are the 6 anterior pituitary hormones, the hormones that control the release and their primary targets, what is a long loop negative feedback for anterior pituitary hormones, for the negative feedback for insulin and parathyroid hormones, what are the actions for hormones. We need to talk about the permissiveness, synergism, and functional antagonism as they apply to hormones. Your endocrine pathologies are the 3 main common or the most common type of endocrine pathologies. Explain how negative feedback can be used to determine the location of a problem. with one gland in a 2 or 3 gated pathway. Hormone evolution explain how comparative endocrinology is very useful for understanding human physiology. And in all of that, you must be knowing all of these basics. Okay, these terms like receptors, peptides, and proteins. What are the differences between endocrine and nervous systems? You'll also know about signal transduction, steroids, and specificity. Now we will talk about the Graves' disease. Okay, this is Marty Feldman showing up using his bulging eyes caused by Graves' disease for your comedic effect. Um, Graves' disease. Okay, the ball slid by the hole and trickled off the green. Another boogie. Ben Crenshaw's golf game was falling apart. A 30-year-old professional had won the Masters tournament only a year ago, but now something wasn't right. He was tired and weak. He had been losing weight and felt hot all the time. He attributed his symptoms to stress, but his family thought otherwise. After urging, he finally saw a physician. The diagnosis? Graves' disease, which is resulting in an overactive thyroid gland. In the course of this study, you will be encountering a different series of questions related to our problem. And at the end of this lesson, you may comment down your suggestions or your answers below on all of these questions. So let's start. Hormones. Okay. At the background, you will see an insulin hormone. Okay, an illustration which is, as you all know, insulin is a protein hormone which is being released in your pancreas. It regulates the metabolism of carbohydrates, proteins, and fats. It does it by preventing the uptake of glucose from blood by liver cells, skeletal muscle cells, and of the fat cells. And you have seen here a disruption of insulin function, which can lead to, as further stated earlier, to diabetes. Okay. What makes a chemical a hormone? Because these are more important thing that we need to understand right now. It goes back in 1905. Okay, that the term hormone was coined from the Greek verb meaning to excite or to arouse. And the traditional definition of a hormone is a chemical which is secreted by a cell or a group of cells into the bloodstream for transport to a distant target. This hormone will exert its effect at a very low concentrations. However, as scientists are learning more about the chemical communication in this body, definition is continually being challenged. Hormones historically have been known since the ancient times. Although the scientific field of endocrinology is relatively young, diseases of your endocrine system have been documented for more than 1,000 years. The evidence of your abnormalities in the endocrine system can be seen from this ancient art. For example, this pre-Columbian statue of a woman shows a mass on her neck. Okay, this mass is an enlarged thyroid gland or goiter, which is a common condition high in the Andes, where the dietary iodine needed to make thyroid hormones was lacking. And we need to understand the historical basis on what this group of people are experiencing before our modern technologies have fully understood, okay, the field of endocrinology. And moving forward, we have here a summary of the hormones according to their location, okay, which glands are secreting them, the hormones, and the primary targets and their main effects. I'm not gonna discuss all of these things, okay. I just have to discuss with you the major locations or the glands of your body that are secreting these hormones: pineal gland, your hypothalamus, your posterior pituitary, the anterior pituitary, thyroid gland, parathyroid gland, thymus, your heart, your liver, stomach and your intestines, pancreas, adrenal cortex, adrenal medulla, kidney, skin, your testes for the male and your ovaries for the female. We have also the adipose tissue and your placenta for pregnant females only. And they are being tagged with these different keys. It could either be their location is a G stands for a gland, C for endocrine cells, and N for neurons. You have also for the hormones, it could either be a peptide, a steroid, or an amino acid derived hormone. And these are their primary targets and their main effects. Accordingly, hormones can be classified to different schemes. Okay, the scheme which is being used in this particular table is a group according to their source. Okay, and a different scheme divides hormones into those who are released and it's being controlled by the brain, and those who was released is not controlled by the brain. In other scheme groups of hormones, according to whether they bind to a G protein coupled receptors or a intracellular receptors and so on, a final scheme will be discussed, okay, further on, which divides the hormones into the three main chemical classes. It could either be a peptide or protein hormone, a steroid hormone, or an amino acid derived hormones or amines. Now, in the clinical connection, the discovery of insulin was brought to us by the disease, we call it diabetes. Your type 1 diabetes mellitus is a metabolic condition which is associated with insulin deficiency and has been known since the ancient times. Until early 1900s, physicians had no meaning or have no means of treating this disease, and patients are invariably dying. It was a series of experimentations that pinpoint the cause of your diabetes. And in 1889, someone from from the University of Strasbourg in France, Oscar Minowski, surgically removed the pancreas from your dogs and noticed that the dogs developed symptoms of your diabetes. And he found that implanting pieces of pancreas in under the dog's skin would prevent your development of your diabetes. And in the year 1921, Frederick Banting and Charles Best in Toronto, Canada, was able to isolate a substance which is from pancreatic extracts that reverse your elevated blood glucose levels. From there, it was a relatively short process until 1922, purified insulin was being used. In a first clinical trial, science had found a treatment for this once fatal disease. And we will talk about in a separate video on how your pancreas, your insulin, your glucagon works, okay, in the pathology and physiology of this disease. Now, in order for us to understand hormones, we need to classify them according to their as stated earlier, your scheme of hormones will be presented in this table. This final scheme will divide the hormones again into the three main classes. We have your steroid, peptide, and your amine hormones, which are tyrosine derivatives. Okay, the peptide or protein hormones are composed of your linked amino acid. Okay, the steroid hormones are all derived from cholesterol, and for amine hormones, these are modifications of every amino acid or single amino acids, either your tryptophan or your tyrosine. So you can see here in the table the synthesis and storage, released from parent cell, transport in blood, half-life, okay, their location of the receptors, response to receptor ligand binding, the general target response, and some of their few examples. Okay, so insulin is a peptide hormone. Okay, for steroid hormones, we have your estrogen, release your ovaries, androgens, and your cortisol. Your catecholamines, the epinephrine or norepinephrine and dopamine, and also your thyroxine from your thyroid hormones. Now, we will look on this peptide hormone synthesis and release, or the processing. So, the peptide hormones are made as large, again, inactive pre-prohormones, which includes a signal sequence, one or more copies of the hormones, and additional peptide fragments. So here, okay, we will try to see how it's being synthesized and how it is being packaged, okay, of these peptide hormones into what we call as the membrane-bound circulatory vesicles, which is very similar to any of other that proteins. So the initial peptide that comes off the ribosome is what we call as your pre-prohormone or an inactive protein. Okay, your pre-prohormones, which contains one or more copies of a peptide hormone, and a signal sequence will direct this one, okay, to it will signal or direct the protein into the lumen of your rough endoplasmic reticulum and any other peptide sequences which may have or may not have any biological activity. So as the inactive pre-hormone moves through, okay, you will see here as it moves through the endoplasmic reticulum. This is your ER, this one. As it moves through your endoplasmic reticulum, the signal sequence is removed and it creates a smaller, still inactive molecule. We call it the prohormone, something like this. Okay, and they will be in the Golgi complex. The prohormone is being packaged into some secretory vesicles. Okay, and this process is what we call as your post-translational modification. As we go look back in your central dogma of biology, you will also learn that there are some post-translational modifications that these polypeptides or proteins are going through before they could have or serving their purpose. And also, once it is being placed inside your transport vesicles, your prohormone will pass through the ER through your Golgi complex, and some of the secretory vesicles will just be containing some of the enzymes, and the prohormone will bud off the Golgi. Now these enzymes will chop the prohormone into one or more active peptides plus additional peptide fragments. Okay, and the secretory vesicle will just release all of its contents by exocytosis into your ECF or the extracellular space. Now, on the last phase, the hormone moves into the circulation for transport to its target. Okay, so this fifth stage here, okay, is also called as the co-secretion phase. Your fifth stage. We have here pre-prohormones, which is an example of this pre-pro TRH, which is a thyrotrophin releasing hormone, which has 6 copies of the 3 amino acid hormone TRH. Okay, the pre-prohormone has again 242 amino acids, which will process into a signal sequence plus other peptide fragments and 6 TRH with 3 amino acids each. Now, your prohormones, which is what an example of prohormone is a pro-opiomelanocortin, the prohormone for ACTH, may contain several peptide sequence with biological activity. So this is your pro-opiomelanocortin, we will process into a peptide fragment, an ACTH, a gamma lipotropin, and a beta endorphin. So lastly, your end of prohormones will process to an active molecule plus some of the peptide fragments. So the peptide chain of insulin's prohormone is folding back on itself with the help of what we call as your disulfide bonds, this S bonds here. And the prohormone will cleave to insulin, this portion here. There are also peptide, your sulfide bonds, disulfide bonds, and also a C-peptide as a product. Now, peptide hormone receptors and your signal transduction. So, again, peptide hormones or this H could not enter their target cells, and they must be combining with different membrane receptors, which are tagged as R, okay, that initiates what we call the signal transduction process. So they will be binding here, and then it will open up an ion channel, this G here, which is a protein receptor. Now it opens up an ion channel, and also it will also give us a cascade of events which will lead to this amplifier enzyme, an AE, to a second messenger system. Okay, your second messenger system will phosphorylate the proteins and making up a cellular response. In response to that, this another receptor here will be activating this tyrosine kinase, a TK, and also all together they will phosphorylate proteins for a cellular response. Most of your hormones or steroid hormones are made in the surface of your adrenal glands or the adrenal cortex, or sometimes in your gonads, your ovaries and the testes. Steroid hormones are not stored in the endocrine cell because of their lipophilic nature. They love fats. Okay, they're fat lovers, and they are made on demand and diffuse out of your endocrine cell. So the first one is your cholesterol. Cholesterol is the parent compound of all the steroid hormones, and we have keys here. Okay, your DHEA or your dehydroepiandrosterone, and this beige color is the intermediate compounds whose names have been omitted for simplicity. For cholesterol, okay, you can also see here an asterisk, which is an each every step is being catalyzed by an enzyme, but only two enzymes are shown in this figure, your aromatase and your 21-hydroxylase. So this is certainly the process. It will be catalyzed into this DHEA into androstenedione, and also your aromatase to produce estrone. Again, your androstenedione will be catalyzed to create testosterone, and aromatase to create estradiol, which are found in your ovaries. Okay, and lastly, it will be catalyzed to create a dihydrotestosterone or the DHT. And on the other hand, your cholesterol will be accessing an intermediate compound here to release progesterone, and progesterone will be catalyzed to a certain intermediate compound, and then an enzyme, the 21-hydroxylase, will be catalyzed into an intermediate compound and then creating a cortisol. And lastly, your progesterone from this pathway will be catalyzed via the 21-hydroxylase, an intermediate compound, catalyzed to form corticosterone, catalyzed to form an intermediate compound, now producing an aldosterone. So they are being classified according to color. Okay, for this one is your from ovaries. Okay, this is for your adrenal cortex and adrenal medulla. Now, steroid hormones act primarily on intracellular receptors. So first is this protein carrier. Okay, since most of your hydrophobic steroids are bound to plasma protein carriers, only unbound hormones will pass through or diffuse into the target cell. So steroid hormones are in the cytoplasm, okay, or your nucleus. So there you go. Your soft surface receptor. Okay, in your 2A, this is steroid hormone binding to a cell surface receptor, a membrane receptor that will be using a second, okay, messenger systems to create rapid responses, rapid cellular responses. On the third phase, this receptor hormone complex will bind to your DNA and activates or represses one or more genes. And number 4, Now, transcription will produce an mRNA. It will cleave off, passing through the nuclear envelope. Now, for translation, it produces a new proteins via passing through this endoplasmic reticulum. This is your steroid hormones. Now, amine hormones, which are derived from the amino acid tyrosine. Now, your tyrosine is the parent amino acids for catecholamines and thyroid hormones. Now, catecholamines are made by modifying the side groups of tyrosine, and your thyroid hormones are created from two tyrosines like this and an iodine atoms. So, amino acid derived amines hormones are some small molecules created from either, again, from tryptophan and your tyrosine. Both are notable from this carbon ring structures, okay, and there are groups. Your pineal gland hormone melatonin, which is being derived from the tryptophan, okay, but the other amino acid derived hormones, your catecholamine and thyroid hormones, are made from your tyrosine. Catecholamines on this orange color are a modification of your single tyrosine molecule, again, and the thyroid hormones will combine tyrosine molecules into an iodine atoms. Despite a common precursor, okay, the two families of these tyrosine-based hormones have little in common. Your catecholamines, such as your epinephrine and your dopamine, are neurohormones. Okay, they are neurohormones which are binding to a cell membrane receptors, the way peptide hormones are doing. And your thyroid hormones in purple are produced by a butterfly-shaped thyroid gland in the neck, which is behaving more like a steroid hormones with an intracellular receptors that activate your genes. Now, in Graves' disease, we continue the problem. Shaped like a butterfly, the thyroid gland straddles the trachea just below your Adam's apple. The thyroid gland concentrates an iodine, an element found in food and most notably as an ingredient added to salt, and combines it with amino acid tyrosine to make two thyroid hormones, which are the thyroxine and the triiodothyronine. These thyroid hormones perform many important functions in your body, which includes the regulation of growth and development, oxygen consumption, and the maintenance of body temperature. The first question goes like this: to which of these three classes of hormones do your thyroid hormones belong? And if a person's diet is low in iodine, predict what happens to the thyroxine production. We will answer that one by submitting or commenting down your answers in this reply section below. Now, for the control of hormone release, we will be learning what are these pathways where the synthesis of hormones are being studied. For the first part, a simple endocrine reflex, which is the parathyroid hormone, and the second one, the multiple pathways for insulin secretion, and they are being powered by negative feedback. So you can also take a look at the figures that are being used, the key colors. We have the stimulus for yellow, the red for the receptor, the violet for the hormone, the blue for the target, and the green one is for the tissue response. This orange neuron is for the sensory neuron, the purple neuron is for the efferent neuron, the red one is for an integrating center, just like a CNS, and the green one is for the systemic response. For the first one, a simple endocrine reflex, an example of that would be the parathyroid hormone. There is a stimulus which decreases the plasma calcium concentration. Now, your parathyroid cell, okay, from the integrating center, will give us a hormone, the parathyroid hormone, passing to the bloodstream. Now, targeting different organs such as your bone and your kidney. And then these are the following tissue response: increase in bone resorption, increase in kidney reabsorption of your calcium, then increase of intestinal absorption of calcium and production of calcitriol in your intestines, and all of these will contribute to an increase of your plasma calcium concentrations. Once it reaches this increase, there is an opposing feedback or a negative feedback that will be hindering the process. Okay, on your right, the multiple path for insulin secretion. Once you have an increase of blood glucose during a meal, okay, there will also be an increase of glucose in lumen. So all of these are again a domino of events or cascade of events. Now, when you eat a meal, there is a stretch receptor in your digestive tract. Now, it will be a sensory neuron which will sending a signal to your CNS, and processing the information to send the efferent neuron signals, okay, to your pancreas. Now, your pancreas as an integrating center will be releasing insulin. They will pass into the bloodstream and go into the target tissue. Now, there is an increase of glucose uptake and utilization. They will be using all of the sugar in your body. Now, they will be decrease blood glucose concentrations. On the other hand, once you eat a meal, there is also a glucose in lumen as a stimulus, which will be felt, okay, by the endocrine cells in your small intestine. Now, they will be releasing a hormone which is a GLP-1. Now, again, same thing with an efferent neuron from the CNS. They will also trigger the pancreas to release insulin for the glucose uptake and the utilization. So this is your next structure, which is your pituitary gland, which is made of two glands with different embryological origins which are being fused during development. It sits on a protected pocket of your bone connected to the brain via your thin stalk. So this is an anterior and posterior view of your pituitary gland. So this is an infundibulum, which is a stalk which connects your pituitary to the brain. This is your sphenoid bone, your posterior pituitary. Okay, posterior pituitary is the extension of a neural tissue, this one, and your anterior pituitary is the true endocrine gland. Okay, your anterior pituitary is the true endocrine gland, which is secreting 6 classic hormones. Neurotransmitters from the hypothalamus will control the release of your anterior pituitary hormones, and the hypothalamic hormones will reach the anterior pituitary through a specialized region in of the circulation which we call as your portal system. Okay, so as we track this process, the first is the neurons synthesizing tropic neurohormones will release them into the capillaries of your portal system. So all of these neurons from your hypothalamus, again releasing, okay, to the portal system, and your portal veins will carry the trophic neurohormones directly to the pituitary, to the anterior pituitary, where they act on your endocrine cells. And the endocrine cells which are found in your anterior pituitary gland, okay, will release the peptide hormones into the second set of capillaries for distribution to the rest of the body. So this vein here will release the target organs, in prolactin to your mammary glands, your GH to the musculoskeletal system, your TSH for the thyroid gland, ACTH for the adrenal cortex and the gonadotrophins, your luteinizing hormone and follicle stimulating hormones, to your gonads. Now, the posterior pituitary is an extension of your brain which is secreting neurohormones which are being made from the hypothalamus. First, your neurohormone is made and packaged in cell body of a neuron. Now they are being transported down the cell, and these are vesicles which containing the neurohormone are stored in your posterior pituitary. Now, for it to be released, okay, there is a vein that will gather all of these and being released into your blood. On the other hand, once they're being released, either this example or your oxytocin and your vasopressin, which are being utilized in mammary glands and the uterus, okay, and the vasopressin, which is responsible in your kidneys for the water reabsorption. So now we are going to talk about the hormones of the hypothalamic pituitary pathway. Your hypothalamus, okay, secretes releasing hormones and inhibiting hormones which will acting on the endocrine cells of the anterior pituitary to influence the secretion of their hormones. So let's use in the homeostasis concept here, okay, and we have to know that the alternate names and abbreviation for the hormones are shown in the table figure, okay, on the next slide. So we have here your hypothalamic hormones in purple, your dopamine, TRH, CRH, somatostatin, GnRH, GHR, and GHRH. So these are the neurons in your hypothalamus which are releasing and inhibiting the hormones into some kind of portal system. So for anterior pituitary hormones, we have prolactin, TSH, ACTH, GH, FSH, and LH. So starts with the dopamine, okay? They will be releasing in your hypothalamus, triggering prolactin release from your anterior pituitary. Now, the non-endocrine targets, which are the breasts, are the tissues for their target response. TRH will be triggering a TSH release in the anterior pituitary, going to the thyroid gland, and your thyroid hormones are being released like T3 and T4 and being brought to the many different tissues. Same thing with your CRH to ACTH, adrenal cortex, cortisol, many tissues. GHRH and somatostatin will be contributing to the release of the growth hormones, going to liver, and insulin-like growth factors, going to many different tissues. And now GnRH for follicle stimulating hormone and luteinizing hormones as gonadotrophins will be released to the endocrine gland cells of your gonads. They will be also releasing androgens to many of the tissues and estrogens to many of the tissues, progesterone. Now, androgens and progesterone specifically for the germ cells of the gonads. So all of these are releasing hormones, okay? The RH or the IH, inhibiting hormones or the releasing hormones. So for anterior pituitary hormones, again, prolactin, a summary, okay, it is an hypothalamic inhibiting hormones of your dopamine, okay, dopamine. Thyrotrophin and TSH is RH, okay. Corticotrophin releasing hormone is an RH, GnRH, which is dominant, and your GnRH is also an RH, and your somatostatin is a growth hormone inhibiting hormone, is a GHIH. This diagram on your left is showing us the growth hormone pathway. So remember that the hormones of this anterior pituitary gland controls so many vital functions that the pituitary is often named okay, called as the master gland in your body. So in general, we can say that the anterior pituitary hormones control metabolism, your growth, and also reproduction. And most of this will be some of the very complex process in your body. One anterior pituitary hormone, for example, is your prolactin. Prolactin, PRL, controls milk production. In females, it's called lactation. So female breast, this prolactin stimulates the milk production. Also, for this diagram, we have here the growth hormone, which is also called as your somatotropin. It affects the metabolism of many tissues in addition to the stimulating hormones production by your liver. So you can see here the liver and producing some of the bones needed. Okay, for instance, for your prolactin and growth hormone, these are the only two anterior pituitary hormones with hypothalamic release inhibiting hormones. Okay, you can see it in the figure, and we will discuss more of it in the preceding slide. So the remaining four anterior pituitary hormones all have other endocrine gland as their primary target, as you all know. Follicle stimulating hormone, the FSH, and the luteinizing hormone, the LH, known collectively as your gonadotropins, as we can they're originally named for their effects to ovaries, but both hormones have effects on the testes as well. Your thyroid stimulating hormone or your thyrotropin controls the synthesis of hormones and secretion of in your thyroid gland. Your adenocorticotrophic hormone, or the ACTH, or the adenocorticotrophin, acts on some certain cells of your adrenal cortex, which controls the synthesis and the release of the steroid hormone cortisol. The cortisol is your stress hormone. We are seeing now two different diagrams, which is negative feedback in some of the complex endocrine pathways. In the first one, in complex endocrine pathways, the hormones of the pathway serve as negative feedback signals. We have here short loop negative feedback, which is occurring in prolactin, growth hormone, and ACTH. On the right side, control pathway for the cortisol secretion. Cortisol is again a steroid hormone, which is secreted by the adrenal cortex, and noted here the ACTH and the CRH. So the pathways in which your anterior pituitary hormones act as trophic hormones are among one of the most complex endocrine reflexes, because they involve three integrating centers. Again, three integrating centers. We have the hypothalamus, your anterior pituitary, and your endocrine gland. So feedback in these very complex pathways follows a different pattern. Instead of the response acting on the negative feedback signal, the hormones themselves are the feedback signal. Okay, we can show it here in this figure. This short signals. Okay, short signals. In your first is hypothalamic pituitary pathways. The dominant form of feedback is long loop negative feedback, where the hormone secreted by the peripheral endocrine gland feeds back, okay, to your anterior pituitary. So it feeds back here in IC2, and it feedbacks also in your hypothalamus. Okay, in this method, okay, it feeds back to suppress the secretion of its anterior pituitary and hypothalamic hormones in pathways with two or three hormones that are being secreted. Okay, the downstream hormone usually feeds back. The downstream hormone, this one, the downstream hormone, it usually feeds back to suppress the hormones that controlled its secretion. And a major exception to long loop negative feedback pattern, okay, an exception for that is the ovarian hormones, which is the estrogen and progesterone, where feedback alternates between positive and negative, and we will discuss that one as we will learn the female hormones before pregnancy. Some of the pituitary hormones also exhibit short loop negative feedback, okay, and ultra short loop feedback. In short loop negative feedback, a pituitary hormone feeds back to decrease the hormone secretion by your hypothalamus. For instance, we have prolactin, a growth hormone, and ACTH. They exhibit all of these short loop negative feedback. in your pituitary and your hypothalamus, in which, again, your hormones will act as either an autocrine or paracrine signal to influence the cell that are being secreted by it. The short loop feedback pathways also are usually secondary to the more significant long loop pathways. Going back to our Graves' disease discussion, the thyroid hormone production is regulated by the thyroid stimulating hormone, the TSH, which is a hormone secreted by the anterior pituitary. The production of TSH is in turn regulated by the neurohormone tryptophan-releasing hormone, which is the TRH from the hypothalamus. The third question is, in a normal person, when thyroid hormone levels in the blood increase, will negative feedback increase or decrease the secretion of TSH? In question 4, in a person with a hyperactive gland that is producing too much thyroid hormone, would you expect the level of TSH to be higher or lower than a normal person? We are now going to the hormone interactions. Okay, hormone interactions. This one will talk about synergism. So if the synergism, it's the combination of two or more hormones, which is resulting in a greater than additive effect. Okay, so this is synergism. If, like, for instance, if one hormone could not exert its effect fully unless a second hormone is present, the second hormone is said to be permissive to the next. So it's like helping, supporting to the first. Okay, if one hormone opposes the action of the other, both of them are okay. In this example, we show here that two or more hormones are interacting at their target, so that the combination yields as a result that is greater than additive. For example, 1 plus 2 has a greater impact than 3. Okay, again, this is synergism. And for our epinephrine and glucagon example, a synergistic reaction would be, for epinephrine alone, this green one, it elevates your blood glucose for around 5 to 100 ml blood. Next would be your glucagon. It elevates also your blood glucose for like 10 mg per 100 ml of blood. Your epinephrine plus glucagon in this sky blue, it elevates your blood glucose for like 22 mg per 100 ml of blood. In other words, the combined effect of these three, okay, is greater than the sum of the effects of two hormones individually. So there are a lot of mechanisms, okay, or cellular mechanisms that underlie the synergistic effects, although it's not clear until now, okay, what is the mechanism between this. But with peptide hormones, synergism is often linked to the overlapping effects on target cell second messenger systems. Okay, and synergism is not really limited to hormones. We have it could happen into like any two or more chemicals in your body, and pharmacologists are developing drugs with synergistic components. For example, the effectiveness of your antibiotic penicillin, which is enhanced by the presence of your clavulanic acid in the same pill. Going back to our Graves' disease, we will continue the discussion. Okay, when Ben Crenshaw was diagnosed with Graves' disease, one form of hyperthyroidism, the goal of the treatment is to reduce the thyroid hormone activity, again because it is hypersecreted. Okay, there is a hypersecretion of your thyroid hormones, and Ben's physician offered him several alternatives, and one treatment involved drugs that prevent this thyroid gland from using iodine, and another treatment was a single dose of radioactive iodine that destroys this thyroid tissue. A third treatment was surgical removal of all or part of the thyroid gland, and Ben elected initially to use the thyroid blocking drug. Several months later, he was given this radioactive iodine. My question is, why is radioactive iodine rather than some other radioactive elements such as cobalt are used to destroy thyroid tissue? Moving ahead, for our endocrine pathologies, okay, these are the diseases in the endocrine system. Diseases of your hormones are usually due to hypersecretion, and symptoms of this hormone deficiency occur when there's too little hormone is being secreted. We call it hyposecretion, and an abnormal tissue responsiveness may result from problems with hormone receptors or the signal transduction pathways. And the primary pathologies arise in the last endocrine gland in a reflex, and a secondary pathology is a problem with your anterior pituitary trophic hormone. We are seeing here, in connection to our endocrine pathologies, and negative feedback from an exogenous hormone. In this example, exogenous cortisol, okay, is being administered as a drug, and it acting as a negative feedback signal, okay, just as cortisol is being produced and how it is being produced within the body, and shutting off the production of your CRH and your ACTH. These are being shut off, okay. Without the trophic nourishing influence of ACTH, the body's own cortisol production shuts down. Okay, if your pituitary remains suppressed and the adrenal cortex is deprived of ACTH long enough, the cells of both glands shrink and lose their ability to manufacture ACTH and cortisol, and the loss of cell mass is what we call as your atrophy, okay, without nourishment. If the cells of the endocrine gland atrophy because of an exogenous hormone administration, they may be very slow or totally unable to begin a normal function. Okay, and with the treatment with this, okay, exogenous hormone is being stopped, again it will be very slow and totally malfunctioning. As you may know, we have the steroid hormones such as the cortisol that can be used to treat the poison ivy and some of the severe allergies. However, when treatment is complete, the dosage must be tapered off to gradually allow the pituitary and adrenal gland to work back up to the normal hormone production. And as a result, packages of steroid pills are being directed to patients which ending treatment to take 6 pills in one day. Okay. After that, and so on, low dose over-the-counter drugs or steroid creams usually do not pose a risk of feedback suppression when used as directed. Now, here we can see here trophic hormone levels will help isolate the source of pathology in hypercortisolism. So we have 3 diagrams here. Primary hypersecretion due to the problem with your adrenal cortex. Secondary hypersecretion due to the pituitary problem, and tertiary hypersecretion due to the hypothalamic problem, which is very rare. Okay, going back, if your pathology, okay, which I think it's a deficient or excess, arises in the endocrine gland in a complex reflex pathway, the problem is considered to be a primary pathology. Okay. For example, if a tumor in your adrenal cortex begins to produce excessive amounts of cortisol, the resulting condition is called primary hypersecretion. If a dysfunction occurs in your anterior pituitary, then it is a secondary pathology. For example, the pituitary is damaged because of head trauma, and ACTH secretion diminishes, and the resulting cortisol deficiency is considered to be a secondary hyposecretion of cortisol. And also, pathologies of hypothalamic trophic hormones will occur infrequently. They would be considered as tertiary hypersecretion or hyposecretion. And how do we diagnose it? This pathologies in your very complex endocrine pathways depends on our understanding of negative feedback in the control pathway. So it shows here, these 3 diagrams here, the showing the 3 possible causes of excess cortisol secretion, and to determine which is the correct etiology or the cause of a disease in a particular patient, the clinician must assess the levels of the three hormones in this particular control pathway. Going back to our Graves' disease again, it is noted that Graves' disease is one form of thyroid gland hyperactivity. And for this reason, people with Graves' disease have elevated thyroxine levels in their blood, and their TSH levels are very low. If levels of TSH are low and thyroxine levels are high, is Graves' disease a primary disorder or a secondary disorder, one that arises as a result of a problem with the anterior pituitary? Now, explain your answer. In this figure question, you're being asked for each condition. We used arrows to indicate whether levels of the three hormones in the pathway will be increased, decreased, or unchanged. Okay, you have to draw the negative feedback loops where it is functional. So how do we do that? First is the hyposecretion from damage to the pituitary, and second is the hyposecretion from atrophy of your adrenal cortex. So for figure A, okay, your CRH will be high, ACTH will be low, cortisol levels will also be low, and no negative feedback loops that are functioning. In letter B, CRH is normal to high, your ACTH is high, cortisol is low, and since there is an absence of negative feedback by cortisol, it increases the trophic hormones. Okay, short loop negative feedback from ACTH may keep the CRH, okay, between these two within the normal range. Thank you. And lastly, as we go ahead with the conclusion of this Graves' disease, researchers have learned that Graves' disease is an autoimmune disorder in which the body fails to recognize its own tissue. In this condition, the body produces antibodies that mimic TSH and bind to the TSH receptor and turning it on. These false signals are fooling your thyroid gland into overproducing thyroid hormone, and more women than men are diagnosed with Graves' disease, perhaps because of the influence of female hormones on thyroid function. Stress and other environmental factors have also been implicated in hyperthyroidism. So question 7 and question 8 goes like this. Antibodies are proteins that bind to the TSH receptor. From that information, what can you conclude about the cellular location of your TSH receptor? And last question, in Graves' disease, why doesn't negative feedback shut off thyroid hormone production before it becomes excessive? Okay, thank you. This last portion of our discussion with the endocrine system is talking about the hormone evolution. Okay, the ability of non-human hormones to work in human was a very critical factor in the birth of endocrinology. And when Best and Banting discovered your insulin in 1921, and the first diabetic patients were treated with that hormone, the insulin was extracted from cow, pig, or sheep pancreases. Okay, before the mid 1980s, slaughterhouses were the major source of insulin for the medical profession. Now, with genetic engineering, the human gene for insulin has been inserted into a bacteria, which then synthesizes the hormone, providing us with a plentiful source of your insulin. Okay, although many hormones have the same function in most vertebrates, a few hormones play a very significant role in the physiology of lower vertebrates and seem to be evolutionary on their way out in humans. Calcitonin, one of the good example of such hormone. It plays a role in calcium metabolism in fishes, but it hasn't apparently no significant influence on daily calcium balance on adult humans. Neither calcitonin deficiency nor calcitonin excess is associated with any pathological conditions or symptoms. Although calcitonin is not a significant hormone in humans, the calcitonin gene does code some of like biologically active proteins, and in the brain, cells process this mRNA from the calcitonin gene to make a peptide known as the calcitonin gene related peptide, or the CGRP, which is acting as a neurotransmitter. The ability of your gene, or one gene, to produce multiple peptides is one of the research which is shifting from genomics, okay, to physiology and proteomics, which is the study of the proteins and their physiological function. One of the good approaches, or to better understand the endocrine structures, okay, that are more important in lower vertebrates, okay, are the vestigial parts in humans. Okay, it means that in humans these structures are present as in minimally functioning glands. For example, in this diagram, you'll see your melanocyte stimulating hormone, which is MSH, from the intermediate lobe of your pituitary, which is controls pigmentation in your amphibians and reptiles. Okay. However, adult humans have only one vestigial intermediate lobe, and normally they do not have measurable levels of MSH in their blood. In the research area comparative endocrinology, okay, the study of endocrinology in non-human organisms has made this very significant contributions to our quest to understand the human body. Many of the models of human physiology are based on the research carried out in fishes, or rats, or frogs, and to name a few. And for example, the pineal gland hormone melatonin was being discovered through research using tadpoles. Okay, many small non-human vertebrates have like short life cycles that facilitate studying aging or reproductive physiology, and genetically altered mice like the transgenic or the knockout mice have provided researchers valuable information about proteomics. Opponents of some of the animal research argue that scientists should not experiment with animals and at all, and should only be using cell cultures instead and computer models. And cell cultures and models are valuable tools and can be helpful for the initial stages of your medical research. But at some point, new drugs and some of the procedures must be tested on intact organisms prior to clinical trials for humans. And responsible scientists follow guidelines and appropriate animal use, and limiting the number of animals that's being killed to the minimum needed to provide the valid data. So I hope you've learned something. In this lecture, we have examined how the endocrine system with its hormones help regulate the slower process in your body, and as you will see, the nervous system takes care of more of the most of the rapid responses which is needed to maintain homeostasis. Okay, this is just like the peak of the melatonin released by the pineal gland during nighttime as you sleep. Okay, it follows your circadian rhythms. So again, thank you so much for taking the time to listen. If you have any questions regarding the lesson and what have you learned, you can comment down your feedback and comments below. Once again, this is Sir Steven, and thanks